I want to talk about Enzymaster, where we want to apply enzyme engineering for innovative and sustainable catalysis in chemical manufacturing. So before I go into detail on our technology, just a short introduction of Enzymaster itself. So Enzymaster was founded in China, in Ningbo, which is just south of Shanghai, in 2013 as Enzymaster Ningbo Bioengineering. Here we have our lab space, our fermentation pilot plant with 2 lines that go up to about 1,000 liters. And also we're working on building up a manufacturing plant for biocatalytic productions, where we then can catalyze reactions in an up to 5 cubic meter scale. And since 2018, we have an international business unit here in Germany. It's Enzymaster Deutschland GmbH, which is located in Düsseldorf. And here we take care of the international business, taking care of the European and North American customers. So as I mentioned, we would like to apply enzymes in catalysis of chemicals. Why do we want to do this? So enzymes have many advantages. We have shorter synthetic routes because we don't need protective group chemistry or any activation chemistry. They also have a very extraordinary selectivity as being chiral molecules themselves. They have high stereoselectivity, also regio and chemoselectivity. Throughout nature, there's also a broad spectrum of transformations which are available. Nature's had a lot of time to develop these, so many different kinds of reactions that can be catalyzed. And also a topic of this event, of course, green chemistry. So we want mild and environmentally friendly reaction conditions. What's the drawback, though? The problem is that natural enzymes are often not industrial applicable. So they're unstable or do not have a high activity on the unnatural substrates. So we try to solve this problem by using enzyme engineering with directed evolution to get the properties which a customer needs to apply in their biocatalytic process, to get these properties for the enzyme. So how do we do this? So we would start from a project idea, a molecule that a customer would like to synthesize with a biocatalytic method, and we would then screen for an enzyme which can do this reaction. So we combine this with computational methods. We screen through any open access databases to find an enzyme which can catalyze the reaction. And if this enzyme also has already the properties which we need for industrial process, we can go into the fermentation development and the industrial process and then have a commercialization of the process. However, as I mentioned, natural enzymes are often not active enough for this, so then we would go into a directed evolution approach. Here we've developed a BioEngine platform, we call it, where we have a bioinformatic platform where we first do a pre-analysis, so we identify hotspots in the enzyme which we want to mutate, which could give us beneficial properties. We screen these in silico, also in the computer, and then also recombine these to then, on the basis of this analysis, create a small library which we then test in the lab, express these enzymes, screen them for their activity and see if we have an improved variant. If we do have the properties that we want, we can then go to the commercialization, but if we still don't have enough activity or stability, we would then go into a new round of evolution. To offer this, we have a comprehensive technology platform where we can cover research and development projects using bioinformatics, high throughput biochemistry, automation to actually carry out the high throughput, on strain engineering. But we combine this with a manufacturing platform so that we can also work on process development, upscaling, fermentation, and manufacturing. So why do we incorporate so much computational navigation? Well, if we look at the classical mutagenesis, using, for example, EP-PCR or site saturation mutagenesis, you're usually only incorporating one mutation at a time into the enzyme. So we're covering only a small space of the sequence space, which is possible, only taking small steps, maybe just reaching a local maximum then of what we could actually achieve. By using computer-aided evolution, we make much bigger jumps. So the vast amount of variants which we can screen in silico covers much more of the space, and also by combining multiple mutations at a time, so 3 to 4 mutations in one enzyme, we can make much bigger jumps and hopefully then reach a global maximum. So with our BioNavigator technology, which is our bioinformatics platform, we can use quantum mechanics to determine activity of the enzymes. We can then screen about 10,000 variants a day. We also use molecular dynamics to simulate protein and environment interactions. There are about 1,000 variants a day possible. And also we can use molecular docking to study inhibition and substrate binding properties. So to give you just some examples of what we've worked on, we have, for example, a transaminase for the synthesis of R-phenyl ethyl amine. This enzyme had a very strong product inhibition, so it was 5 grams per liter already inhibited. And also the isopropylamine, the amine donor, which we needed to incorporate the amine group, also deactivated the enzyme. So at 2 molar, which is a high concentration, but we want this to push the reaction, the enzyme was also inactivated by this. And by using molecular dynamic studies, we could then simulate the interactions of the solvent and the product with the enzyme, and we can see in the red that there are destabilizing parts in the enzyme, which are then we target for the mutations. And after 10 rounds of evolution, we had an enzyme which was a lot improved. So we have over 300 gram per liter product was tolerated. So going from 5 to 300, which is a great improvement. And also the stability was increased. So at 2 molar isopropylamine, the enzyme was stable for 180 hours. Another example is the ketoreductase, which we used for the synthesis of R1,3-butanediol. This enzyme was not very firmly stable, so this is always a disadvantage. If you want to do a partial purification of the enzyme with a heat treatment, then you usually lose a lot of your activity. Also, it had a very low conversion, and low substrate loading at 60 gram per liter was not very well tolerated by the enzyme. Also, studying the structure of the enzyme, we found positions which were destabilizing, and by targeting these and mutating these, in just 3 rounds of evolution, we could get the stability to last for 2 hours at 85 degrees, and also the conversion was increased to near quantitative, and 400 grams per liter substrate loading is quite high, so you can get multiple metric ton production of this chemical. And just to give you an example of time which is saved by using these in silico methods and not actually having to do everything in the lab. If you did a fully combinatorial NNK library of 6 positions, you would take your library size would be about 1 billion, and it would take over 10,000 years. So nobody was going to do this in the lab. But by using these bioinformatic methods, we can then pre-select mutations which we want to incorporate. And this is reduced then down to 2,000 variants. And this can be screened in 8 days. Another example, just to show how we want to make synthesis more greener. You can see the synthesis of chloramphenicol, an antibiotic. It takes many dangerous and toxic chemicals to produce this compound. And you can also see in the red arrows that these are actually reactions which don't contribute to the end product. They're all just protective group chemistry or activation chemistry. And so these could actually potentially, with enzymes which are more selective, could be avoided. So we wanted to catalyze the aldol addition in an enantioselective selective way so that we could actually reduce the synthesis by 5 steps and avoid protecting and activation group chemistry. So in this example, the aldolase firstly started out with not a very high stereoselectivity though. So the diastereomeric excess was only about 50%. And with a 10% conversion in 8 hours, this was not very good performing enzyme. So we then took this into evolution by modeling the substrate in a transition state combined to covalently bound to the coenzyme PLP in the enzyme. We could then find out which positions were important to mutate so that we get the right conformation in the binding of the pocket and therefore get the right diastereomeric selectivity. And in just 9 rounds of evolution, we then increased the diastereomeric excess to 90%, also with a conversion of over 80%, and also a high substrate loading could also be achieved at 200 grams per liter. And just as I mentioned at the beginning, there's many transformations which can actually be catalyzed by enzymes. So we have many different enzyme classes which can catalyze different reactions. We can create chiral amines, for example, chiral alcohols. So these are important reactions and for pharmaceutical synthesis, for example. We can create carbon bonds by the aldol reaction, and this also stereoselectively, or also do a decarboxylation. So these are just some examples, and also examples which we've worked with, but there are still many possibilities which are unexplored really. And to give some examples that enzyme catalysis can really be applied on a very large scale, we have some commercialized products which we produce. So for example, for pharma and food, we can produce beta-alanine. We can produce this up to 100 to 150 metric tons a month. So it's a very large scale. This is all catalyzed by enzymes, or for example, airborne-free butanediol, which can be produced in up to 50 metric tons, which can be used in pharma, food, or cosmetics. An example of a chiral building block would be L-tertiary leucine. This is applied in, for example, HIV medicine, in atazanavir medicine, and also chiral building blocks, for example, naphthyl ethyl amine. These can all be done in multiple ton scales, so quite a large scale. And this brings me to the end of the talk. So just to summarize how Enzymaster tries to help companies incorporate sustainable enzyme catalysis in their chemical manufacturing. So first we would identify an enzyme to find an enzyme that can actually catalyze the reaction which is desired. We then engineer this with directed evolution methods to optimize the enzyme's activity, stability. We can even go for enzyme immobilization, which would then make a workup more easier after the reaction. We can then go into process development, optimizing scale-up of the reactions, or work on downstream processing, also on enzyme fermentation development. And in the end, reaching a commercial process so that we can then produce our chemicals on a more sustainable way. And with that, I'd conclude. Thank you for your attention. If there are any questions, I'll gladly try to answer these.
So thank you very much, Dr. Hayes, for this very interesting talk and the insight into the world of engineered enzymes. For the audience, again, you can use the Mentimeter for questions, and I will start with something I popped up here. So my first question, maybe you brought it in between the lines. So how long does it take if you have an idea and then produce it in silico and engineer your production pathway up to the demonstration scale, for example? So how many days, weeks, years do you need to do this?
So this is a question that we often find because customers are, of course, working on deadlines. So it's very difficult to say. So sometimes we can do a screening project and find an enzyme which is suitable for the process. So that would maybe take 2 or 3 months. But if we have to go into an evolution, one round of evolution takes about 5 to 6 weeks. And we can never really guarantee that after 1 or 2 rounds we will already have the properties which are desired, of course. So we have to think about, will we get these properties? Is this enzyme well known? Is there much known in the literature about this? Or are there already mutations which are beneficial, which we can already incorporate? So this all takes time, and of course analytics is also a problem. So if the analytic of the product takes long and we're trying to screen 2,000 variants, it takes quite a long time to screen all these for each round. Typically, projects can last up to half a year, but they can go longer. But an optimal way would be, of course, just 1 round of evolution, and then that could be into about 2 months, and we have a process. But then you have to go in the upscaling, and hopefully that will then work as well. So,
and that's a good anchor for my next questions, the upscaling. So what are the main challenges when you are coming from in silico research to the fermentation or to the biotech pathways?
It varies strongly depending on the enzyme. Sometimes there's no problem going upscaling, but if you, for example, take oxidoreductases like monooxygenases, which incorporate hydroxyl groups into compounds, they usually need oxygen in a high amount, so you have to really stir the reaction well, and at a higher scale this then gets a bit more difficult, and then it's more difficult to scale this up. I think what's being worked on a lot is immobilizing, of course, the enzyme, so that you could then maybe work in a flow kind of style, that you just flow the the substrate through the column, for example, and the reaction takes place and the product comes at the end. So this, I think, is something that continuous methods are going to be a big thing in the future. But it really depends on the enzymes if there are problems with the upscaling or not.
Okay, so we have a question from the audience. Are you also doing some research and development in Düsseldorf at the university, or is the focus fully on business and marketing? So for the university, sorry.
Our main focus is on the business and on the chemical manufacturing companies so that we can there incorporate these green methods. But we do also, we're open to helping research groups with our methods. So if there's some funding which we can apply for with the university groups, for example, then we're open to doing this as well and then try to help with our bioinformatic methods, for example. But actually, we are only an office in Germany, so our research is actually all carried out in China in the mother company. Okay.
And for the piloting or for the upscaling, do you work with other partners, or do you do your upscaling by your own?
Partly on our own, partly with partners, so it depends on the scale. I mentioned we have a fermentation pilot plant. There we can go up to a fermentation scale of 1,000 liters. Anything that goes above this, we work with partners then together, and also for the chemical manufacturing. So we are at the moment working, building up a new chemical manufacturing plant where we can then carry out reactions to 5 cubic meter scale. But if it goes over this, then we work with partners together.
Okay, and then another one from the audience. Is your technology focusing mainly on small and simple molecules, or can this be applied to more complex components as well?
I would say at the moment we're quite focused on small molecules because I think these enzymes are more been in focus in the past, also in the literature. But the limit's basically open. So if there are good enzymes which can catalyze on complex systems, and we would also do this. So it's at the moment we're focused on small molecules, but I think the limit is open what the future can bring.
Okay, question by myself that goes a bit in the same direction and was already also for Professor Axmann. Do you concentrate more on some kind of large-scale production? You had some base chemicals as example, or do you more concentrate on fine chemicals or specialties for pharmaceuticals or something like this? Where are the opportunities in these ways?
We do both. So in our mother company in China is more focusing on the large-scale production, so this would be more of bulk chemicals. But we also do fine chemicals, but we can also do small-scale reactions. So if there are just a few kilograms needed, then this is also possible. So it depends on what the customer wants. But often the customer, which we find at the moment, is preferring to have the enzyme developed and then do the manufacturing themselves. So we then just work on the enzyme, can also act as an enzyme supplier, and then the company actually carries out the manufacturing themselves.
Okay. Then I have two very specific questions from the audience. Were metalloenzymes already produced in an active state and efficient manner?
Well, I mean, yes. Yes.
Okay.
So, I mean, for example, keto reductases are also sometimes metalloenzymes. So they have a metal ion, zinc, for example, in the center. So this could be a metalloenzyme, but yes.
Okay, and the second one, is your approach limited to molecules with specific functional groups, or is it also possible to synthesize medium and long chain paraffins or olefins?
That would also be possible, yes. So it doesn't have to be small molecules. So if we find enzymes that work well on these long chain olefins, for example, then there's no problem in engineering this enzyme as well. So yeah.